So in fact, this is really meant more as a course than a research seminar. It's fine, because so I gave the seminar the other day. I hope you could listen. So the seminar was more about what we do. This is more a sort of survey of what exists, to give you an idea. I try to make it not too boring in the sense that I will give examples that apply to certain things and not only techniques. It's okay, and I will go through my applications for different questions. Okay, so yeah, so in general, that field of microfabrication for cell biology has been developing for quite now a lot of time, but I think it's really in the last 10 years that you start to see it a lot in cell biology papers. Okay, but in fact it exists for longer than that. So the four things I've put in there are so different types of approaches. So one is micropatterning, so that applies more particularly to that course, I mean what we are doing in the practical course. And what I want a bit to underline, to show in that part is that there are really thousands of ways of doing micropatterns, and they apply more or less to different questions. Then you have some cells which cannot adhere. All cells are not mammalian, adherent cells or things like that. So for example, disease cells, and you might still want to change their shape, for example. And for that, you will need 3D structures, and the simplest ones are microwells. Microwells are also very useful, I mean, for many things. Then another level of tools you can are generally called microfluidics. So when you're talking about microfluidics, it means that in these structures you are flowing things. It's not only that you change the shape of cells, but you also control the liquid that's around the cell. And so the interest of microfluidics is that you have laminar flows, so you can control liquids in a very precise way. Here you just see a cell which is running in a channel. I showed you that we're using these microchannels for cells. And these channels also have some little structures. If they are biased, you can bias the migration of cells. And finally, let's say the most complex type of things is when you go in a 3D structures, microfabricated 3D structures. So this one here is still pretty simple. It's just that you have beads and you put some material that you can polymerize around the beads, and then you dissolve the beads, so you have a sort of honeycomb material. And these kind of materials are used now quite a lot by people who want to do tissue engineering. So I will end up with examples of people who try to do blood vessels on a chip or lung on a chip or these kind of things. So what you want to control with these devices are the growth medium. So this is usually microfluidics, and you want to control it spatially, gradients, or temporally. You want to control the surface for adhesive cells. So this is more like micropatterning or wells. And also all this can be done in materials which have specific mechanical properties, and you might want to have to go to 3D and combine all that. So one, again, I think I said it the other day, the real important thing is to stay simple, in the sense that your device fabrication should be much simpler than your biological experiment. Otherwise, you will do device fabrication and not biology. And that's my point of view. I mean, of course, if you have good collaborators who do all the device fabrication work for you, maybe it's good, but it's also nice to, I mean, the type of device I will talk about is really device you can make on your own, and which are, I mean, the principle is that they are simple. This is summarizing the fabrication technique, which is all the different things, they all rely on this kind of technique. You have a material which is sensitive to UV, and you have a mask, and you shine UV through your mask, and the regions which are illuminated with UV, they change properties, and so either they get destabilized or they get stabilized, and then you make a sort of mold. And this mold can be used to mold whatever you want, elastomers, hydrogels, and with that you can make stamps. So for those who make the who follow my session, they have seen these stamps, and you can stamp protein. Or you can also use them to make chambers or wells or this kind of thing. But at the end, it's always that. So it's this sort of photolithography which has been developed for microelectronics and which was transferred for biology. I must say, really, the people who really popularized that is the lab of George Whitesides of Harvard University, and they call this kind of technique soft lithography, using materials like PDMS, which are biocompatible. PDMS have lots of disadvantages and advantages. The advantage is that it is permeable to gas, so your cells will be oxygenated if you are in a PDMS chamber. Problem is that it's hydrophobic, and if you're using small molecule inhibitors, they might diffuse inside the PDMS. So there are some solutions for that. Yeah, so basically that's it. So it's the most used material, let's say. So first, let's talk about micropatterns. Here you see a cell. In fact, it's not a cell, it's a large number of cells which have been averaged on top of each other, and that was possible because they all have the same shape. So they're all adhering on a pattern which is like that. And so here you see 75 images of centrosomes, which are all averaged here, 75 images of nuclei. Okay, and so this is this idea of that was promoted by Manuel Terry of having a sort of average cell. That's one of the use of the micropattern. So the main question is after how you make so. So for some people who have not seen that, so the principle of the pattern is this universal principle I told about this morning, is that you have a region where cells bind and the rest where they do not bind. Okay, so when you put cells on that, so people will see that in a few hours, I think. When they find a region where they can stick, they stick on it, and the others, they remain floating. So once you have your cells stuck on your patterns, you can flush the other ones, and you end up with this sort of surface with your cells in a given shape and in a sort of array. So I was mentioning in a practical term, one thing which is a bit hard to optimize when you want to have single cell micropatterns is to have one cell on each pattern and not 2 or 3 or 0. And you cannot hope, I think, more than 50% of your pattern with a single cell. It's hard to get better than that. And usually, when you are just saying that and you put cells, if you see all your patterns filled, you can be sure you have on most of your patterns more than a single cell. So usually, it's better to be on the lower density side where you have, let's say, half of your patterns without cells, and then usually the other half as a single cell. So that's what it looks like when a cell is spreading on a pattern viewed by RSEM microscopy. So it's a type of microscopy which shows you the contact between the cell and the substrate. When it is in contact, it is dark like that. And you see how the contact spreads on the pattern. So the cell is following these branches of adhesion. So there are several interesting things in there. One is that quite soon the cell will reinforce adhesions on the ends of the pattern. You see, here, here, and here, because in fact it is generating fibers between these ends, stress fibers, which are pulling, and pulling on these adhesions is reinforcing the adhesions. Okay. It's quite nice. In fact, RICM is a very nice tool that is used quite a lot, but not enough. I think you can even be quantitative in terms of distance between the cell and the substrate. Here you start to see the cell is pressing on its nucleus, so you see its nucleus here. It's just pressing on it. And there it is non-adhesive region, and I think it was one of the main finding which was really unexpected. I mean, it was not planned, this finding from Manuel Terry, that cells, if you put them on such pattern, they would spread on the non-adhesive region, and then they would have a non-adhesive edge. And this non-adhesive edge is, you see, it was first kind of curved, and then it became tensed, because there is a fiber here which is pulling between these two ends, and this makes a very nice stress fiber between these ends. So you see here is a fibronectin adhesion and this very nice trace fiber here in actin. And so that was a bit I mean, seeing that by chance, I must say, really completely by chance. Then when you see this principle, you can design all kinds of shapes to control where the cell will form fibers. So if you have a T, you will have 2 fibers. If you have a Y, you will have 3 fibers, these kind of things. And you can play with cell polarity. The next application, which was, I think, very interesting, was to move these micropatterns to in vitro assays with the exact same principle of having regions which are sticky not for cells but for proteins. And then you can have actin nucleators, and you can form these very nice patterns of actin just by printing the nucleators. So here it's an example with cells, which was, I think, for many people in cell biology, was really the paper which was, I mean, I was really just starting my PhD when it came out. And that was really as a demonstration of the interest of following this sort of methods. The patterns existed before, but that was really a striking example. So here it's a paper from Ingber, and Christopher Chen is the first author. I think you probably know these people. And so here the idea was, so when cells spread on substrate, it was known for some time that so if cells are on small, for example, if they don't have a lot of adhesion on small patterns, they will die. And when they can spread, they would survive. But it was not clear if what they needed was to engage their integrin receptors on fibronectin, or what they needed was to be spread. And so this experiment is just to show you a bit how you can means the kind of idea you can have in mind while you're using micropatterns is, for example, here you want to distinguish between these two possibilities, and so you design here a pattern where here this intermediate, so here you know they will die, here you know they survive, and here this pattern is giving you the answer because the cell is spread on an area which is as large as that one, but it has a total adhesion surface which is as small as that one. Okay? And what you find is that, in fact, on this one, the cells survive, which means that what was important was the spreading. Okay? And then, of course, you've understood, there have been a lot of other work to understand why it is the case, but I think it's a good example of what you can do. So here you have other examples of using clever ways of using patterns. Here you have a ring which is either centered or off-centered, and they have looked at where the cells proliferate. The idea was you can see that proliferation occurs mostly at the edge of the pattern, and the idea was to say that cells which are at the edge are under more tension, and that promotes proliferation. Here they have patterned alternative stripes of two types of adhesion molecules. One is collagen, so it's extracellular matrix, and the other is cadherin, for example, and you can see how cells would form these two different types of adhesions on the two different types of stripes. So this is a bit to show. I think I don't have to convince you that you can do lots of things with patterns, because these are all kind of examples. But when you say, okay, I have a question, I want to make patterns. The problem is how you choose how you make your patterns, because if you start to just type micro patterns, you will find hundreds of papers, and it's not very easy to know how to choose one. So I just review a few existing techniques. So this technique is, I think, the technique that people used, very first technique that people used to make patterns, because it's really directly photolithography. So it's called photolithography and liftoff. So what you do is what I showed you before. You will cover your cover slip usually, so your glass substrate, with this material which is sensitive to UV. Then you use a mask, and for example, here in that case, you have the two cases as usual. In that case, you stabilize the part which is illuminated with UV, and so when you solubilize the rest, you will end up with your glass, which is masked by these little islands. Then you cover with your molecule of interest, and then you lift off the remaining resin. Usually you need to use a solvent to do that. And so you end up with a substrate where you have this protein which is just deposited at certain places. So this is a very robust method. It works with pretty much everything. The problem is, as you see, you have a large number of steps, and this has to go through, usually be done in a clean room. You need a spin coater to put your resist. You need all kinds of solvents to remove it. And for each cover slip, it will be quite painful to go through all that. But in case nothing else works, this will always work, I think. And here you see a nice example where little dots have been patterned. And depending on the distance between dots, cells will behave differently. Here they look like cells on a regular type of substrate. Okay, they don't particularly care about the dots. But here, if the dots are a bit far away, they start to align and form some more regular shapes because they start to make stress fibers between the dots. So this is a technique which was introduced, in fact, by Welle, Peter Bauer, and before we used it. They used it mostly on polystyrene. And what we introduced is a combination of this UV technique and the use of polylysine PEG. So really what we wanted to do is to find a way to make it as simple as possible and that you could do it on your bench. And so here is you have a surface. First step is to coat it with PEG, destroy the PEG with the deep UV through a mask. Then you just incubate it with your proteins, and then you plate your cells. So here is just basically an incubation step to put the PEG, UV 5 minutes, another incubation step, and you're done. So it's very nice, very simple, but it's not very robust. I mean, in many cases it will not work because, for example, the PLL PEG will not protect enough for the type of protein you want to put here, for example, or because your particular type of cell is very adherent and it will be able to go on the PLL PEG, or for other reasons like that. Most of the case it works, and it needs little adjustment to make it a bit more robust, but yeah, for us so far, so now we are using only that. So I think it's a good guess to try that as a first example. So the things I've been showing you before, they produce kind of, okay, you have a mask, you produce your pattern on all your surface. But it's not very flexible. Each time you want another pattern, you have to make either another stamp or to buy another mask. So there was a whole set of techniques which were developed, which were more versatile in how you make different shapes. So here, it was developed before that paper, but in that paper is a good example. So it's basically always the same. You have a surface which is covered by something which is non-adhesive. Here they use PVA, but it could be PEG. It's the same with PEG or with any other molecule. And the idea here is to use a laser to burn this layer of non-adhesive material. And here they use a 2-photon laser. I think, so it's very nice. It's a bit of an overkill because when you have such it's very expensive material, I mean, maybe you don't want to use it to make patterns. It works very well with a simple UV laser. And use a very cheap like diode, you know, these UV diodes. It's really good enough to make your patterns. And then you need some sort of, it can be here they use AOM, but it can be galvanometric, whatever, mirrors to move your laser around and make the pattern. So basically here what you do is you just draw any sort of design on your computer, and it will the moving mirrors will move the laser around, and they will burn the pattern you want. An interesting application is that you can do sequential patterning of different proteins. And here I must it's just note that the other technique you can also do that. It comes from the fact that very often when you put a protein on a pattern and you saturate the pattern, if you make another pattern and you put another protein, it will not go on the first pattern because your first pattern is saturated. Okay, and there are little tricks to make it work really as well as you can, but it's simpler than you might think at the first place. So the main limitation of that technique is that when you're patterning, you are patterning very locally. you are patterning just under where your laser can, and if you want to, and then you have to move to make another pattern and move to make. So it's very nice if you have a cell and you want to turn something around your cell and follow what that cell is doing. If you want to have a whole cover slip with patterns, you can forget about it. I mean, it will take half a day to pattern your cover slip. Okay. That one I show because I think it's a very nice idea. Although I never saw really people using it, but it's just to show you how simple it can be to make patterns. So these people have just coupled a projector, like that one, to an objective. Okay, and so now I could, with this method, I could just project this slide, for example, on a cover slip, and make pattern which correspond to this slide. So the idea is just that these projectors, they can send a UV light. Okay, and you can use this light to create patterns. I think this is of course not commercial. It takes a little bit of doing the right thing to make it work, but I think it's a good way to have very versatile patterning method. So this is the most classic patterning method, which is the micro-contact printing. So here you start with the photolithography as before, and you make a stamp, and then you just stamp your protein on your cell. So this is the reason why it's so popular. I think is that once you get your stamp, stamping. I mean, it's just. I mean, it takes some practice, but it takes nothing else. People have done it. I mean, you just put your drop of protein on your stamp and you stamp it. Yeah. So I think it's still probably the most widely used technique. The only limitation is to get the stamp, and the big technical limitation is the reproducibility of your stamp. I mean, there you have people who are very good at doing it, people who are less good, and you can develop tons of little tricks. You will never get something very homogeneous on large surfaces and very reproducible. But often it's good enough for cell biology. Okay, then we move a bit out of, so I think you've seen the most classical type of techniques and what you can do with them in terms of their advantages and limitations. Here there is a bit more exotic thing, so just, of course, it seems obvious, but you have just to think about it. You don't have necessarily to print proteins, you can print organelles. So here, people have printed centrosomes, and from these centrosomes you can grow microtubules. It's not very clean, huh? And in fact, I didn't see any other paper using it, so I think it's probably not so useful. So in fact, there is now another paper it's very recent, so it's not in my course, I'm sorry, which was by Manuel Thierry again, where he was printing, in fact, little microtubule seeds. And you can make very beautiful asters with that. Just micropatterning little microtubule seeds. Here you can print cells also. You can directly print your cell instead of printing the protein. So here in this example they have been printing a bacteria. So here you have an array of bacteria. So each little dot is at the beginning a single bacteria which will make a colony. So it's very nice because you can have a homogeneous colony size and follow how they grow individually. Another technique which has its advantages, so we've moved out of the really typical micropatterning technique, is a stencil. So stencil is, you have to make this stencil, it's a bit like you make your stamps, but it's sort of inverted stamp. So it's instead of having pillars, it has holes. And through these holes, you can plate cells. So you see that example, for example, it's nice because it's sort of going through a multi-well plate system. And in each well, you have these little holes, so you can print your proteins. And it's also nice here because for co-culture. Because you can print you don't have to have a non-adhesive and an adhesive substrate. You put your cells in the holes, you remove the stencils, and you put your other cells around. Okay. And you have co-culture. I would say there are many limitations to that technique. The first one is to make the stencil is not so easy because you see it's a very thin layer. It would break. So here they found the trick of putting the stencil at the bottom of the wells. Yeah, I think it's a bit tricky, and you are really limited in the size of the holes you can make here. For single cells, it would basically not work, the stencil technique. It works for a large population of cells. So the next level is to have patterns which can be more dynamic. So this is using the UV tool, the UV laser tool, so just burning. So basically the same as we saw before, except that you implement a new pattern while the cell is already on the pattern. For example, here you had a cell in green, which was in this V pattern. Then you you burn a line here, and the cell will spread. You see it here. It's on the V pattern. So yeah, it's a bit different. So here they've made first a line on top here. So the cell finds a line and spreads on the top line. Then a line on the bottom, and the cell spreads off the bottom, and you go from a triangle to a rectangle. Okay. So here I think the idea is that here you have a cell with primarily a stress fiber here, and you end up with a cell with two stress fibers here. Yeah, so you can do very nice things here. Another technique and so this is simple, but it's this single cell level. If you want to switch at the population level, you need to switch the property of your substrate globally. And so here it's a method which is called electrochemical desorption, where your repellent protein, the PEG, instead of being bound in a permanent way to your substrate, is bound with a link which is sensitive to tension, voltage. Yeah, that's because in French you would say tension, but it's voltage in English. And so if you apply, so if you link your substrate, it has to be a metal or conductive substrate. You apply a voltage, a difference of tension. You will remove the PEG layer. Okay, and so the cell here, which was stuck with PEG around, it can start to move out of this pattern. So in that example, the idea was to check whether the shape of the cell would really determine its direction of motion. So you see, often you see cells which have this shape and they are moving that way. But here it's on the pattern, so it's not moving. But what happens if you remove the non-adhesive part, and you see that indeed it is moving that way. So this is a technique which is based on photochemistry. So here the molecules that you use to repel, so this is sort of, in fact, it's a molecule which is hydrophobic on which you put BSA. It has one part which is sensitive to UV. So if you limit with 365, it will break here, and you end up with this molecule on which you can put fibronectin, and then the cells will bind on that. And so like that, you can do that. You have your group of cells, you eliminate a round here, and these cells will switch to this other round. Or here you have cells, and you eliminate here, and the cells will switch to here. So I realized that in fact here I forgot to add a slide on the new techniques that we developed with the click chemistry that I showed you this morning. But the paper is out in Advanced Material if you want to check, and all that you can do also with this click chemistry, with different things. That's the same idea. You switch the properties of the substrate. So here, just to show that your substrate itself can be dynamic. So here it's just pattern on PDMS. And so if you pull on it, you see these cells with these two stress fibers, you pull on it and you get it elongated and it reinforces these stress fibers. Here it's pattern on polyacrylamide, so the cell will deform the pattern. I think here the idea is, I mean, it's not been used so much, but I think it will turn out to be very interesting for traction force microscopy, instead of having cells which are just spread on the pattern, to have cells which have a defined adhesive pattern. So here you have a pattern printing of polyacrylamide, so the cell is pulling on it, so it's deformed. And when you inhibit contractility, it relaxes and you see the initial pattern, okay, which is. And so, in fact, comparing this pattern and this pattern, you can know the force that the cell was exerting here. And you can have force maps like that, which are very average on many cells. So this is a technique you have been trained, I mean, some of you have tried last week, I think. Developed by Christopher Chen and by also Benoît Ladoux in parallel, this micropost. So why do I mention it here? Is because here this technique you measure forces, so this is another way of measuring forces by the cells this time pulling on the post. Yeah, so this is in general I would say here you switch to techniques where, in my opinion, it's a bit less accessible than a simple micropattern. But if you can team with people who know how to make that, it's also very useful. This is the same thing but with large posts. Just to mention that you can, it's also very interesting to access the forces produced by groups of cells instead of single cells. I really like that example. It's like a small muscle that you assemble on these things, and in fact you rely for that to work on the fact that the cells themselves will rearrange the fibers in this little chamber, and the fibers will get caught on the posts here, and so when they will contract, they will bring the posts together. That's Christopher Chen lab. So now, here you've seen like single cell micropatterning. It was all based on techniques which work only if the cells can stick to the substrate. A second set of tools can be used even when cells are not adhesive cells. And the simplest of this tool is a microwell. Again, I take that example just to show you. So just with something as simple as a microwell, you can already do a lot of things. And I will go through some examples. Here, in these wells, you can see a yeast cell. It's a Pombi cell, which because it's growing normally as a straight cell, but when it encounters the walls, it will bend because it has not enough force in that case to push the wall. And you can learn a lot about the mechanics of the wall of these cells just using these simple chambers. This is a work by Nicolas Mink in the lab of Red Chang. So starting from the smaller scale, here it's an example with bacteria. And I give you all these examples. I hope it's not too painful, because it's true that going through this is a bit painful. But it's a bit to open your show you that with all these techniques, the only limit is what you want to do actually. So it doesn't matter if you're working with bacteria, yeast, tissues. Okay, this thing applies to all these scales. So that's examples I will show you. So here it's a bacteria. And so for bacteria, they have been using the usual growth medium, which is agarose, instead of using PDMS. And except your agarose, you just pour on your mold, and you get agarose with little holes. Then you have your drop of cells, and you close it with a glass slide, and you just look through it. And your cells are trapped in these little holes. So it cannot be more simple. And then for example, here your bacteria will grow, but it's trapped in there. So instead of going straight, it will start to grow in circles. So then you form a bacterium which is not straight anymore, but has a shape a bit like a helix, like that. Here it has this sort of shape, for example. Okay, you can see it starts like that, and as it grows, it's bending. So why would you want to do that? In fact, in there they don't do very much with it, but there are two interesting applications. One which they do in there is to try to ask what happens if these cells are swimming. What happens when they swim and they have a different shape? Okay, so it's how they use it. But it's been used since also for very interesting questions about the growth, how these cells grow, and where they insert new material along their length. Okay? Because this cell you can take out of the wells, and you can see how the shape evolves when it's no more constrained, and it's just growing, but from not the original shape that it should have. Whoop. I don't know what happened. Something happened. Here is the example I gave before with yeast cells. And so simply wells, a drop of cells in their liquid. You push simply with the cover slip. You see here it's before, so the cells are out of the well. You push a little bit and up, they are in the well. And then you can watch them grow. You can measure forces. So here's so there I show the work of Nicolas. We've done a similar work in parallel. It was published a bit before, 2008, using microchannels. Here the idea is that these cells, normally they are straight. Here you see the microtubules in green. And they have this very stereotypical microtubular organization which run along the length of the cell. And one question was, what happens if now it's not straight anymore? And you can see that the microtubules reorganize as you would expect for stiff bundles, that they will go where they have to be less curved. But then that has interesting consequences, which are that instead of touching the tips, they touch the side. And in this system, polarity of these cells is determined by where the microtubules touch the wall. So here, normally, they will go only from the tip, but here, because you've bent the cell, it will start to form a new polarity here, and it can start to grow from there. And you can induce a branching of your cell like that. So it was a way to show that how shape feedbacks some cytoskeleton organization, which can itself feedback on shape. Here it's the same device, but used to measure forces. So you see the cell, it's different cells, and here what's changed is the stiffness of the material outside. So if it's very soft, the cell can deform it completely, and if it's stiff, it cannot deform it much, and it bends. Okay? And so with that, you can look at the deformation of the chamber as a function of the Young modulus of the PDMS that you're using, and from that you can deduce the force it takes to buckle the cell. And from this buckling force, you can deduce the Young modulus of the cell wall. I think in fact, it's interesting. I like a lot this. I think it's a very simple. In fact, I have students do that as a practical course in a master degree, because I think it's very easy to show that you can measure forces on very small organisms with a simple way. And Nicolas mentioned to me that in fact there is even a much simpler way to do that, which is just to do osmotic shocks. And you look at the deformation of the cell as a function of osmotic shock, and you get the same number. It's a bit more fancy to do it with microwells. So this is another. So now we're getting to bigger cells, sea urchin cells. But you see, it's just, again, the same thing. It's interesting that Nicolas, he's been using these microwells for several years in Fred Chang's lab, answering a lot of interesting questions through his postdoc, all with these microwells, basically. And you've seen that making microwells is very easy. So here the idea is just like the yeast cell, except that instead of pressing a yeast in the well, you press a fertilized egg, so it's a sea urchin egg, and you can get eggs of all kinds of shapes. And there the question is, when the egg is in a different shape, where will it divide? So if it's round, it has no particular axis, but it will cut in two. What happens when it's not round? And there is an old rule, which is called the Hartwig rule, which predicts that normally the cell should divide perpendicularly to its long axis, and so it has equal volume on both sides. And so you can see here there is a prediction of the Hartwig rule in yellow, the yellow line, and where it actually cuts, and it's pretty good. There are some cases where the cases where it's not good, in fact, are rather cases where the Hartwig law is a bit ambiguous. So you cannot really tell what it should be. But in most cases, it's good. You have to add that it should be a symmetry axis, I think, so you get rid of this error. And the next question was, okay, now that it looks good, but why does it work so well? And Nicolas worked out a model to explain how the spindle finds the long axis in these cells. So this is, again, another application of these wells, this time for stem cells. So this is a work from the lab of Matthieu. And stem cells, for stem cells here, they use hydrogels mostly. So you just mold your hydrogel with your mold, and you have hydrogels. And for some reason, it turns out that stem cells, they prefer to grow in these microwells and on a dish where they are alone in a large amount of liquid. To me, it's not yet clear why it is the case, but it seems to be the case that they survive better in these microwells. It could be that they secrete some things that get more concentrated in these micro volumes. And then you can work on your microwells to make them a bit more to modulate the adhesion. For example, here is a nice trick to get the bottom of the wells adhesive with some adhesion molecules. So the simple trick is that instead of using a mold, which is so this PDMS mold, you will ink your mold. So you have fibronectin, for example, at the tip here. And when you use that mold to produce your microwells, this fibronectin will transfer to the bottom of the wells. And then the advantage here is you can make hydrogels with different stiffness, and you get, and there is also an effect on the way you can amplify these stem cells depending on the substrate, the stiffness of this substrate, and the success of transplantation of these cells. Here it was the same idea, except that here it's hematopoietic cells, and you want to differentiate them in microwells. And the question they were asking there is from Max Krummel lab, was the question of, do the cells need to be touching each other, or do you need simply to have a high density, but they don't need to touch each other to differentiate properly? Kind of typical question you would ask with these systems. So here is the follow-up a bit on that one. Now that you have grown your stem cells on these microwells, you want to put them back in the mice, and the question is how efficient will be the transplantation. And here is as a function of the Young modulus of the wells that you've been using. So that's also from Matthias Lutels and Ellen Blau. I think it's Ellen Blau. Yeah, you can correct me if that's not it. Okay, so it seems to work very well. Somehow, from talking with Matthias Luther, it's not so clear why it works. I think for me it was not always very clear why it works so well. It seems to work. Okay. So now that you've seen these microwells, very simple, lots of application. Here you've controlled basically the shape of the cell, how they adhere to the substrate, but there is another important parameter, which is the content of the liquid that surrounds the cell. Yeah. And the content of that liquid you can want to control locally and also to change with time. And usually this is done with this set of techniques which is called microfluidics. So when you're using microfluidics, you need some liquid to flow in your system. And sometimes it's not compatible with the fact that you want to keep the cells in the system also. So one solution which is used in that paper from the lab of Stan Leibler is to separate the part where you have the cells and the part where you have the flow. And so here you have microgrooves in which these bacteria have been plated. And then you have a layer of cellulose, and on top of that you have a PDMS with a microchannel in which you can flow whatever you want. This kind of device now, they are commercial. You can find them commercial. They work pretty well. I must say that the assembly is not always completely trivial, but it works quite well for yeast and for bacteria. So I like this example because it led to a lot of very nice science afterwards. You can follow, you can look at all the follow-up on this story. But basically, the idea was that you plate bacteria here, so you see all the single bacteria, and you let them grow. As they grow, they will form, they will grow in these stripes, so they will grow as long filaments. Okay, and you can know their growth rate just by measuring the length of these filaments. And then you put an antibiotic, and you kill them all. In fact, not completely all. As you see, this one was not killed, and when you put back your growth medium, it grows, and it's a survivor. And what is very interesting is if you look before you put your antibiotic, this survivor was not growing. Okay? So this cell was in a non-growing cell, and is the only one which survived the antibiotic. And this concept is very interesting because if you think of that cell, if there was no antibiotic, it would have been completely diluted in the population. So in terms of evolution of fitness, it's a very low fit cell. But if you have an accident like an antibiotic, it's the only one to survive. Because why? Because this antibiotic is killing growing cells only. Cells which do not grow are not dead. And so what's important is that in fact this cell is genetically identical to the others. It's just that there is in the let's say the growth machinery of these cells, there is a switch which will switch of the cell in this sort of dormant state at random. And these dormant cells, they will be the ones which survive. And so you can make a whole theory on that, on the fact that you have, in fact, in a population of cells, you have stochastic switch between the states in which the cells are. And some of these states might be of lower fitness for your actual environment. But if the environment changes, suddenly it can be of much higher stiffness than the other cells. And so it's a way to survive, for example, unexpected changes in the environment. And the theory that Steinleibler developed is that if you want to know what is the fraction of cells that should be in each of these states, it should correspond to the probability of this state to become favorable in the natural history of the cells. Okay, so here it turns out that these kind of cells, they happen, I don't remember, in fact, they happen at, I think it's 10 minus to the 7 or something like that, very, very low proportion, but they are responsible for the resistance, so they are quite important. So this is just the same thing, but with yeast cells instead of bacteria in the same type of chamber, a sandwich type. And these studies are also very nice. I mean, they are looking at how cells grow. One point that you can make here, which is important, is also that in this device, you also have nice imaging of this population, and you can do automatic tracking of what the cells do because they grow on a single plane. So here you just saw, like I switched to one medium and I switch back to another, but you can also do that periodically. And in these studies, I think the important concept which was introduced, so they were both at the same time, is they are very similar. This is Van Udenhaden lab, and this is Pascal Hersan. In fact, I forgot the last author name. From Harvard University. And here the important concept is that is really a reverse engineering approach. You have a cell, okay, and the cell is responding to a signal. So here is a simple, the biological process is rather simple. You have a factor which is nuclear, and when you put salt on your cells, the factor, which is cytoplasmic, sorry, and when you put salt on your cells as a response to the salt, this factor gets into the nucleus. Okay? And when you remove the salt, the factor gets back into the cytoplasm. So the readout is the nuclear cytoplasmic ratio of this factor, which gets in the nucleus and out of the nucleus. And now you want to know more about the dynamics of the signal transduction cascade, which is inside the cell. You want to know characteristic times and things like that. Imagine, in fact, this could be just an electronic circuit which has a response. How would you do that with an electronic circuit? You would just trigger, test it with periodic impulses of different frequencies. Okay? And so that's what you can do with microfluidics, because instead of being just one step of salt, you can put multiple steps and you can vary the frequency of these steps, okay? And so it's the frequency response of the system that you get, okay? The driven system by the periodic stimulus, and then you get the response of the system, and you get what's important is the amplitude of the response and the phase shift of the response. And from that, you can deduce some properties of the system. I think at least for me, it was, I think, the first time in these papers I saw this sort of approach implemented for signal transduction. And I think it's very powerful. Here, it's another example of what people have done with microfluidics. What you've seen before is just signals and high frequency control, potentially, of signals. Here, it's multiplexing. So the other interest of microfluidics is parallelizing the experiments. So what you have is the same type of channels you've seen before, except there are many of them. Okay? And in each of these channels, you can put a different strain of bacteria, and you can go through your different strains and image them. And so here in that case, it was a sort of large-scale study of protein content of bacteria, like how many copy number of the protein for lots of different proteins. So it's a kind of the parallelization of the experiments. So I think this is truly an important aspect of microfluidics. It is not necessarily the most easy to implement. Because when you start to parallelize, to have a lot of channels in parallel, it means you have to be able to control all of these channels. Okay? So you can imagine the number of tubes that are coming out of such a device. I would personally not do it, but in some labs there are really specialists of this kind of thing. So they can do it. Okay, so that was, I don't know why there is a separation with oh yeah, so here it's going to mammalian cells. This chamber I found very useful, we use it a lot. It was initially devised, so it's the lab of Jeon, and so it's Taylor et al. Paper, and it was initially designed to separate the body of neurons and the axonal termination. And the way to do that is you have two big channels like that, and between these big channels you have very tiny channels, and only the axonal, so only the growth cone and the axons can move in these channels. And if the distance is enough, so here you see these tiny channels in which the axon will grow. And so if it's small, you will have both axons and dendrites which can cross, but if the distance is large enough, only the axons will cross. And so on the other side, you will have just axonal termination. And it's a kind of basic unit, I think, to start to engineer axonal networks, neural networks, sorry, with different types of neurons. And people now are using that a lot. It's also very useful in general when you want to be able to exchange the mediums on both sides and have, for example, some things in between in the small channels. So we're using it a lot also for cell migration, for example. So this is what it looks like if you look at the mold that you would use for these channels. You have the big channels and the small channels. And in fact, so I also give that example because in terms of fabrication, it is very easy to make. It looks like it's tricky, it's very easy. In fact, what you do is you have simply a mask with lines, and you print these lines on a thin layer of photoresist, and that will give you your small lines here. Okay? And they don't have to be just that length, they can be much larger. And then you come with a second mask which has these big channels on it, and you put a second layer, thick layer, and you just put your second mask at 90 degrees from your first small lines. So you don't need any particular alignment, and you get that. So it's in fact interesting that it's in fact very simple. So this is, I think, the opposite. This is very complicated. I just show it to show that people can go you can go as far as your imagination and your, I would say, your will to implement hard technical systems goes. And so in that case, all these things you see is, so it's a microfluidic chamber with electrodes. And then you can culture neurons in there, and you can do electrophysiology on the neurons in the chips. I've not followed that field so much. I don't know if it went very far or if it was just to play with things. So this example, so then when you're starting to work with microfluidics, the problem often is to control the pressure in the device. And so you have sophisticated way of doing it with devices which give you very fine pressure control. But for example, when you're running this sort of channel, let's imagine that here it's completely separated, like here, okay? And you have this small channel in between. If you're flowing things here, if you have a very small difference of pressure on both sides, you will start to have flows in your channel here. Okay? And that would kind of be bad for what you want to do, in particular if you want to generate gradients in the small channels. And so you have different tricks, and these tricks I like because it's kind of automatic, you don't have to do anything special. It's just that you bring the two channels together here for a short distance, and so the pressure equilibrates here. And because it's lamellar flows, they don't mix so much. And with a little distance, so the concentration re-equilibrates, you get what you want. It's a nice trick, the kind of small trick that makes the device easy to use. So in this paper, they used it to generate gradients for neutrophile migration. So here you see the gradient of attractant in green, and the neutrophils in the channels. What is nice here is that you can see that, so these channels, if I remember, they are 10 microns in width and 3 in height, and the neutrophil completely fills the channel. So if you have a dye on one side and another on the other side, you can see that the dye does not diffuse on the other side of the neutrophil. And it can be quite interesting to, for example, put a drug just on the front and not on the back or this kind of thing. Very useful device. So this is what I've shown during my talk the other day. So for cell migration in general, you might want to use this simple type of devices to disentangle different contribution of the microenvironment, confinement, constriction, rugosity, the fact that it is not smooth, elasticity or flow. All that can be done in simple microfluidic devices. So this I've shown the other day, so I will not show. So we are getting to the last part, and I will finish rapidly, is when you want to put things together to a higher degree, a higher level of integration of different controls. And all the devices I've shown you before are types of devices, for most of them, that I've been implementing in my lab and using with, I must say, quite a lot of success in terms of getting biology done with them. So I'm confident that micropatterns, microwells, microchannels, you can do very nice biology with it. Okay, it's simple, and you can already control a lot of parameters. These integrated devices I've not used. There are more and more of them in the literature. I think for some of them, maybe there are interesting things to do, but I think the future will tell if really they are really helpful or not. But I will show you so you can know that they exist. So this is a kind of principle. I don't think that any device like that exists yet, but it is one of these integrated devices is what people call lab on a chip. Basically, they would want to do everything you do in several steps on your bench in a single chip. Okay? The idea being that you can go to much lower volume, you can be higher throughput or this kind of thing. So, for example, each single of these parts have already been achieved, and people are working on putting all of them in one chip. So you separate your cells, so you separate them, then you segregate them in different types, and on one side you would do biochemical analysis, and the other side you would just image them. I must say, maybe it will come to a point where it is actually more useful than making them in different steps. As it is now, I think it is easier to do it in separated steps than trying to integrate them. My opinion. So this is a classical example of the super complex chip with all the tubings. And here what you do is in all of these little chambers, you control different concentration of different reagents in time, and you have little, each of these chambers is like a little culture chamber with you, where you have so different experiments, and you can have explore a sort of parameter space with that, like having several quantitative parameters which are changed with time. In the space. So this is all kind of things you find in if you go in if you read journals like Lab on a Chip. Typically integrative biology or this kind of thing. People are doing all these crazy systems where, so here you culture single cells in little ways. So this one, I must say, I like quite a lot. It's a clever way to trap single cells in, and then when you flow things, each cell will be in its trap. And the principle is that this trap has a little hole, so the flow goes through the trap, so brings the cell in the trap, but when the cell is in the trap, it blocks the little hole, so the flow goes around. And like that, you can really fill each trap with a single cell. And in fact, the device is not so complicated to fabricate. I think it's quite nice. Okay, and so another approach of like higher level integration is to use these sort of tools to try to go to the level of the tissue and not only the single cell. So this is one of the, everybody knows that example, I think, at least in the field. And I think the reason why it appears so nice is that it's also because it stayed quite simple. So what they wanted to mimic here is exchange between gas air and liquid, which would be blood. And with 2 layers of cells which you find in the lung, which is the epithelium and the endothelium. And so, and between these 2 layers, you have little holes which help so things to circulate at the molecular level. And so this is very simple. They take 1 layer of PDMS which has holes, and on one side they culture the epithelium, and the other side the endothelium. And then they add another aspect of lung, which is the fact that when you breathe, you are pulling on your tissues. Okay? And if you want to assay that, you want to pull on this, and for that you have these 2 side chambers in which you can control the pressure, and so you can pull on the central part like that. And I never tried to make it myself, but it looks pretty simple. It's still just two sort. It's 1 layer sandwiched between 2 kind of regular channels. You can see it like 1 layer, 2 channels. So it seems pretty simple, and that I like a lot also because I'm working with immune cells. So you can, on one side, you can put bacteria, and they will go on the epithelium. On the other side, you can flow immune cells like neutrophils, and they would stick to the bottom layer, go through the hole, and take the bacteria. So you can see it here. You have the bacteria, the neutrophil, which sticks below, and somehow uptakes the bacteria. And so you can have a sort of model of infection, lung infection. Okay. So some people have even proposed to go to organism on a chip. So this organism is quite simplified, but it still has a liver of different parts. I think it's more importantly than the device itself is the idea behind it. The idea is that you, as I just showed you for the lung, where there was just a single function of the lung which was reproduced in the device, which was the exchange between gas and liquid, okay, and maybe infection. You can try to do the same for different type of tissues where you want to assay 1 function. So in general, but so that's I think is interesting for each tissue. In particular, I think I have an example after here for blood vessels. But the concept behind putting them all together is not entirely clear for me that it's useful. But I think that the idea is that you can test drugs. It's a big idea behind that, is that when you test drugs on cells, you are testing something which is very far from the physiology of the body. And so you want to have a system to, for example, put a drug here, and you can see if the drug affects more the lung or the liver or whatever, or other tissues, if you can get it to flow through all these different devices. Okay. It's more the principle, and it's done that afterwards. And so when you have these channels, you can just grow endothelial cells to coat the channel walls. And then in the hydrogel, you can put other types of cells, like your tissue cell. And here you even propose that you have tumor cells which are there. So that people know how to do, and that is just like for you photopolymerize some hydrogels with some cells inside. And here the idea is that the tumor cells will produce some factors which will attract the growth of the blood vessels to vascularize the tumor. And so if you can study that in vitro, you can also try to make drugs to prevent that. So these are different ways of making, as I mentioned before, making some sort of tissue cell-laden hydrogels. So here you have, this is one of the techniques where you just have, for example, you have these microcapillary, you mold your stuff around, you remove the capillary, and you have a hole in the hydrogel. Okay. I think for here again, I don't have the most recent examples. I like a lot a recent example. I think it's from Christopher Chen lab. I think it's a Nature material. You can find it, where he's using sugar fibers, electro-spun sugar fibers. So really like barbapapa, in fact. You know barbapapa, you know this thing or not, or it's just French thing. Is a thing that that kids love. You have a wooden stick, and you have something that rotates and melting sugar, and you put your stick like that, and you have a big bowl of pink sugar stuff. And so with the same machine, you can make a sort of barbapapa like that, and you can dip it in an hydrogel and with cells, and then you have an hydrogel full of these little fibers, and then you just dissolve them. Sugar you dissolve in water, and you get a sort of 3D tissue with capillaries inside. I think it's quite fun. And now people know how to control fiber formation with electrospun techniques. So that's, I have not the slide for that, but this is the idea. And so this is the paper from the lab of Abe Struck to make these blood capillaries in hydrogel. I think it's very nice that now it's quite convincing that you can learn interesting things from these devices. And I'm done. Any question from this large overview? No.
Yeah, I have a question. So when you start, in any, for example, the last case, in any system like this, there are many scales.
Yeah.
The organ there are scales. And the attempt in these is to try and confine your observation to one set of scale. Is that the...
No, I think so. I would say there are different directions that are taken with this microfabrication field. So some people want to control one parameter of the environment. That's more what we are doing, and that's often, it can be at a single cell or multiple cell level, but basically you want to have quantitative control of your environment. Okay, and then you have all kinds of tools for the microfluidics, microstructure, whatever. Then there are people who want, I would say, and then maybe close to that also, if you are talking about an organ, what you want to reproduce is one organ function that you want to study. So that's what you want to get in your device. So if it's for one cell, it can be cell migration, but if it's for one organ, it can be gas exchange, or filtration of things, okay. So and there you do fundamental research on that. Then there is another type of things that people try to do is to use these devices for drug discovery. Okay, so there it's less a question of knowing how it works, but it is you have a certain thing that's going on your chip, a certain function which is already quite high level. For example, in these blood vessels, formation of sprouting of blood vessels, and then you use that as a way to test some drugs, for example, or do some screening that you could not do in a whole organism. Okay? So this is another direction, and there is a third direction, which I think is more really tissue engineering. Some people believe, and in some cases I think it's already achieved, that they can reproduce, they can grow tissues in vitro. And to grow a tissue, you need to, you can think of several approaches to grow a tissue. You can just rely on the cells themselves, that they already know how to make a tissue, but you need to help them be in the right environment to do that. So you need to control the environment. Another way is to really be more like an architect and really direct the way the cells are. And you want to print the cells, that people are doing 3D printing of cells or things like that. And here the goal is not really to understand anything. I think at the end is maybe later on you can use these kind of fabricated tissues to understand something. But there is really a goal to make tissues just for replacement, I think, for medical application. If you can make a muscle, so it's already done for skin, but people are trying to improve the quality of the skin they do, for example. And for all the organs that you might want to replace, it would be cool to be able to culture these organs. So there is that kind of also, for I've seen people making a lot of these scaffolds. So you have a sort of scaffold, you want the cells to be happy to survive in this scaffold and to achieve a certain function, and then you want to implant this scaffold in the body, for example. It can be for bone, for bladder, for muscle, it can be for heart. So nobody has proposed that for the brain, maybe if you want to change your brain. This kind of idea. And I mean, I must say the most crazy one I've heard, I don't know if it's crazy or just a bit frightening, or I don't know, but at least for people who like food, like French people, it's frightening. Some people want to make food like that, like the biosteak, you know, the steak that you make out of an hydrogel with cells inside. And instead of having an animal from which you make your steak, you make your steak directly. So you don't have to kill the animal, it's true, but I mean...
It doesn't taste good.
I don't know, it doesn't exist. Ah, it exists already? It's true? No. Okay. So I know people invest a lot of money in that. I mean, there is obviously a whole interest of the food industry for this kind of thing. I mean, it's kind of... I think on the way, even if your goal is a bit crazy like that, on the way you can learn a lot of things on tissue organization, things like that.
So in the example of the blood test, can you actually get the different cell types to interact? I mean, so pericytes are outside.
Yeah, some people, you can add pericytes or not, or just endothelial cells. So some people have done that to show that pericytes are important to stabilize the vessels or something like that. What do you mean to interact? Yeah. You're mentioning these cells here. So this will be, I think here, yeah. So they would be in the hydrogel. Here it's not empty, it's hydrogel filled with cells. Okay. So I guess maybe it's not easy to get the pericytes just on the sides like that, but I mean maybe you could imagine some methods to do it. What's interesting is this kind of sprouting events, how you make a new blood vessel. People are studying that in actual animals, but you know it's always, at least for me, my personal interest in these things is that when you see this very nice process in the animal, you can make genetics to remove a gradient, or you can image your gradients. Very often there will still be some ambiguity about the causality of the different things, I think. When you can reproduce that on a well-controlled system and you can really change the different parameters, you can start to really prove the causality of the process, in the process. That's, okay. No questions from students. No. Let's go back to make micropatterns.